Okay, so chapter 5, we are talking about the structure and function of large biomolecules. And so there are these 4 biomolecules that we are interested in. These are the carbohydrates, the proteins, the lipids, and the nucleic acids. So we're going to go through briefly each of these 4 biomolecules in chapter 5, talk a little bit about each one. And we'll learn more about these as we go through the rest of the semester, but this is just to introduce you to each of these 4 types of big biomolecules. Okay, so first of all, we need to get a little bit of definitions, some vocabulary under our belt. So a polymer, when we talk about polymers, we are talking about long molecules consisting of molecules that are similar that we refer to as building blocks. So we're talking taking many, many small things together and putting it into a big thing, and we call that big thing a polymer. When we think about the building blocks that make up those polymers, we refer to those as monomers. So remember your prefixes here. So poly is going to refer to many. So that refers to the many small building blocks that we take together. And mono, remember, is going to be one. So we take each individual monomer. So if our squares here are our monomers, we would link those together to make a big polymer. So this whole thing here would be our polymer. Each individual square would be a monomer. So we are going to be using those terms a lot as we go through this chapter, so you need to be sure that you understand what they mean and what the differences are. So our 4 molecules of life, again, here they are. They're carbohydrates, our proteins, our lipids, and our nucleic acids. And each one has a monomer. So the monomer of carbohydrates is simple sugars. The monomer of proteins are amino acids. The monomer of lipids is a little bit tricky. We're going to say that it's fatty acids because the type of lipids that we are talking about are made up of fatty acids, but there are other lipids that are not made up of fatty acids. But for our purposes, we're going to say that the monomer of lipids is fatty acids. And for nucleic acids, the monomer is nucleotides. So we'll go through these individually as we go through the rest of the chapter. Okay, so in order to make these polymers, we have to carry out chemical reactions that allow us to take the monomers and put them together to make a polymer. So if you remember, we had our little monomer squares here, and we linked those things. We drew bonds between them, or linking them, to form our polymer, this one big thing here. So the types of reactions that we carry out, that we use to link small molecules to make a large molecule, are called condensation reactions. So the condensation reaction is taking small things and putting them together to make a big thing. These types of reactions require energy to form the molecular bonds, and water is a byproduct of these reactions. So we'll take a look at what it looks like on the next slide. Okay, so here we have small monomers. So I'm going to kind of draw a box around each of the individual little monomers here. So we have 1, 2, 3 monomers that we are going to take and put together, form bonds between, to make one big polymer. So the way that we do that is we have, these are enzyme catalyzed reactions. So an enzyme is going to come in here, it's going to attack these monomers at these hydroxyl groups. So remember that back from chapter 3. So these are hydroxyl groups, and that's where the reaction is going to take place. So you can see when the enzyme comes in and attacks these hydroxyl groups, we lose water. So we lose 2 atoms of hydrogen and 1 atom of oxygen, and that is released, and what's left behind is our oxygen that is involved in the bond formation between our 2 monomers. So water is a byproduct, and we get the bond being formed with the remaining oxygen between the monomers to form the polymer. These are condensation reactions. We also, in biology, are oftentimes taking polymers. So here's our big polymer. And we oftentimes break those things down back into the individual monomers. So these are called hydrolysis reactions. So here we are breaking large molecules back down in their small molecules. In these types of reactions, energy is released because when we break a bond, we release energy, but we require water in order to carry out these reactions. So we have to add water to our reaction for this to happen. So if we're looking here, again, now we have our big monomer here that we just made in the previous slide. So there's our monomer, or I'm sorry, there's our polymer. This is a polymer here. It's a big, large molecule. So here's our polymer, and we want to break it back down into its 1, 2, 3 monomers. So we are going to have to add water. So in this case, water is going to be one of our reactants or our starting materials. So we have to add water in order to do that. And we're going to break those things back down into their into its monomers. So remember, one of the big key here is these reactions are energy releasing. So we are going to be releasing energy when we carry out hydrolysis reactions. All right, so let's take a look at carbohydrates. That's the first type of polymer that we want to look at. Carbohydrates are made up of simple sugars. So simple sugars are the monomers of carbohydrates. Simple sugars are your sweet hydrophilic substances. So remember, hydrophilic means they dissolve in water. We know that if we were to take some sugar, put it in a glass of water, it would dissolve. Those are our simple sugars. So this is glucose, fructose, the types of monomers that make up table sugars like sucrose that we eat. So we can have just one of those simple sugars, and that's called our monomer. So this molecule right here is glucose. That's one of our most important monomeric simple sugars that we will talk about in biology. We can have short chains of those monomers. So when we take anywhere from 2 to 99 of those monomers and put them together into a polymeric form, we refer to that as an oligosaccharide. When we have 100 or more of our simple sugars in a big long chain, we refer to those as polysaccharides. And so we're going to talk about a couple of different polysaccharides that are important to us as biologists. Saccharides simply just means sugar. So we're talking about oligosugars or polysugars, but we use the term saccharide. So oligosaccharides, remember, are 2 to 99 chains. Polysaccharides are 100 or more. Cellulose is one really important polysaccharide. This is very important to plants. We find it in plant cell walls, and it's what gives those cell walls their shape, their integrity, and allows the plant to not lose water due to evaporation. We also have starch. This is another important polysaccharide. Starch is a glucose storage molecule that we find in plants. Again, this is the way plants store their glucose. And if you look at the differences between those two polymers, between cellulose and starch, you can see that starch is it's a little curvier, a little windier, whereas cellulose, if you think back, was just these big, long, straight chains of glucose. Cellulose has a little bit more susceptibility to degradation. I'm sorry, starch has a little bit more susceptibility to degradation, and that's because we want to be able to break down starch when we need glucose. When we need to use glucose as a source of energy, we need to be able to attack this molecule and break it down. Cellulose, on the other hand, remember, that's what makes up our cell wall, that's what gives our cells integrity. We would not want to be able to easily break down cellulose. So the structure of these two polysaccharides is directly related to their function. The last polysaccharide that I want to talk about is glycogen. So glycogen is an energy storage in animals. So this is the way that animals store glucose. Okay, so plants do it via starch, animals do it via glycogen. You can see, consistent with the starch, that glycogen is susceptible to those enzymatic reactions. We have lots of ways that we could break down glycogen, and that's good for us. So basically what happens is if you go a few meals without eating, let's say you skip breakfast and you skip lunch, your cells are going to need some energy. They're going to need that glucose in order to drive the processes that they need to do. They're not getting it through your food because you haven't eaten. And so the first thing they're going to do is go to their glycogen stores and start breaking down glycogen so that we can get these monomers of glucose so that our cells can carry out the processes that they need to carry out. Remember, we are taking here a polymer and breaking it down into its monomers. So that is that hydrolysis reaction that we just talked about. All right, so let's talk about lipids next. Lipids, and it says don't dissolve in water. I'm going to put a caveat on that and say they mostly do not dissolve in water. So we'll talk about some lipids that actually do have a portion of them that dissolves in water, but for the most part, lipids are hydrophobic, meaning they do not dissolve in water. They do serve as energy reservoirs long term. So these are not the ones that we're going to break down first and we haven't eaten for a couple hours. Remember, that's glycogen that we just talked about, but lipids can serve as energy reserves if, say, we haven't eaten in 3 days or 4 days. Then we're going to start going to those lipid reserves and breaking down those lipids in order to get the energy that our cells need. We also find lipids in cell membranes, so we'll talk a lot about those lipids. Lipids can serve as hormones within our cells. We won't talk so much about those. There are a couple different types of lipids. So you have the fats. These are your fatty acids and your triacylglycerides that you probably have heard about from a nutrition standpoint. We also have phospholipids. These are the ones that we see in cell membranes. And then lastly, we have our waxes and our sterols. We're actually not going to talk about those, so you are not going to be responsible for the waxes and the sterols for this class. But we will talk about the fats and the phospholipids. All right, so let's talk about fatty acids. Fatty acids always have that long carbon backbone. So remember, we talked about this back in chapter 4, I think, when we talked about how carbon was a versatile bonder. We talked about the different types of backbones. So here we have a straight chain hydrocarbon backbone, so all made of carbon and hydrogen. Here we have one with a double bond, so we get that double bond in there, which gives it a kink. Here we have one with a couple of double bonds, so every time we have a double bond, we get a little kink in the chain. But all of these are straight chain carbon molecules that serve for that backbone. So fatty acids have those long carbon backbones. They also have the carboxyl group. So remember those back from chapter 3. So these are the carboxyl groups. They give acidic properties to whatever molecule they're attached to, which is why they are called fatty acids. So that's your COOH, so a carbon, 2 oxygens, and a hydrogen. These carbon backbones can be what we call unsaturated, which indicates that they have a double bond, or they can be saturated, which indicates that all the bonds are single. So this first carbon backbone here, that one would be saturated, so all the bonds are single. These two here would be unsaturated. So you've probably heard of saturated fatty acids versus unsaturated fatty acids from a nutritional standpoint, and that's what we're talking about, whether or not those carbon chains have double bonds. Unsaturated fatty acids are going to be liquid at room temperature. These are typically going to be healthier for you. The saturated fatty acids are the ones that are going to be solid at room temperature, and these are going to typically be the ones that you want to avoid in your diet. All right, so let's look at the triacylglycerides. Again, this is probably something that you've heard about from a nutritional standpoint. So we typically find triacylglycerides in things like butter, lard, and oil. So basically, a triacylglyceride is a glycerol molecule, which we see here. So you're not required to know the structure of that, but that molecule is glycerol, plus 3 fatty acids. So here we have fatty acid 1, fatty acid 2, and fatty acid 3. So we are taking small things here and putting them together to make a big thing. So this is a condensation reaction that we are carrying out here. So we create a bond, or we make a bond between the glycerol and the fatty acid. So there's our bond. So we now have this big thing here, which is all connected, and that is a triacyl that's a triglyceride. So we take a glycerol molecule plus 3 fatty acids, and that gives us a triglyceride. These are really important lipids to us from a nutritional standpoint. We find these in lots of our foods, and our body has to be able to break them down and use them in a way that is efficient for our cells. The other type of lipid that I want to talk about are the phospholipids. These are the major components of cell membranes. So this is what makes up your cell membranes, are entirely made of a phospholipid bilayer. So when we talk about cell membranes, we are talking about something called a bilayer, which is 2 layers of these phospholipids. We'll talk about why that is in a second. So here we have our long chain fatty acids. Okay, so these are hydrophobic. So they do not want to interact with water. They're very nonpolar. They're not going to dissolve in water. That's called the hydrophobic tail of a phospholipid. We also have, though, a hydrophilic head. So this is polar. It's hydrophilic, meaning it will interact with water, it will dissolve in water, and these 2 parts come together to make up these phospholipids. So you can see we have a phosphate group right here in the middle of that polar head, and that's why we refer to these as phospholipids. So remember what we talked about in chapter 2, that almost all of the inside of your cells is water and almost all of the outside of your cells is water. So we want to have cell membranes that have parts that they can interact with water. So remember, the inside of our cells are water, the outside of our cells are water. So in order to do that, we need these hydrophilic heads on both the inside of our cell and the outside of our cell. So if we just had one layer of a phospholipid, where this was my hydrophilic head and these are my hydrophobic tails, and if this was our membrane right here, we would be okay on the outside where we had mostly water because our water would interact with our hydrophilic head. But on the inside of our cell, this is our membrane right here. So on the inside of our cell, we would be in big trouble because we have hydrophobic tails there that don't want to interact with water. So in order to combat that, what your cells do is they create a bilayer. So they put another layer of phospholipids in here on the inside. Now we have a hydrophilic head that is happy to interact with the water inside our cell, and our hydrophobic tails are squished in between. Okay, so they kind of push the water out. They don't have to interact with the water. So you can see that here in this picture. I'll draw a line right down the middle. So we have bilayer number 1 up here interacting with water outside the cell, and we have layer number 2 down here interacting with the water inside the cell. So hopefully that makes sense for you.